Understanding the migration biology of the monarch butterfly in the Pacific Northwest while providing an avocation for prison inmates or incarcerated people. So everybody knows about the monarch. How much you know, I guess, depends on what you've read and what you know. But basically, this is the sort of map that is usually shown when showing the migration patterns of the monarch. And so we know a lot about the eastern population. And conservative estimates used to be 100 million. I don't know about now because the population is declining. But the point is that our population in the west is a fraction of that, maybe 1 million compared to 100 million. So the vast majority are in the east, and we know about their movements because of the extensive tagging. And I hope you all saw the movie, the Seattle, whatever it was, the IMAX theatre thing, which documented Fred Urquhart, and his tagging program that over the decades has demonstrated this movement to Mexico and back again. And even this year, lots of tagged monarchs have been found. Actually, about 150 a week ago, a couple of people from Southwest Arizona Monarch Research Group went to Mexico and they came up with 150 tags. One actually dated back to Fred Urquhart at Toronto. We think it's one of his tags, where he used to put a tag on the forewing and fold it over. So it's been in Mexico at that time. And what happens in Mexico, getting sidetracked, but the people there that live near the colonies go up there in the summer and find the wings on the floor, on the ground, and if they find a tag, they keep it and wait for an American to turn up next year and sell it to them. And so some people have been hanging on to tags for decades apparently. But that's the only way of getting the tags back. And so these women this year got 150, all from a range of different years, because they got the word around. I mean, internet and everything, you'd think it wouldn't be the same, but down there it's very limited communication, and you have to go there one day and spread the word, and then go back 2 weeks later, and then everybody's there with their tags. Whereas normally people just go once and then leave, and so the tags never get bought out.
What's the going price for
tags? $5. $5 a tag, which to them is a lot of money apparently. And they pay the children $3 in the summer to go looking for the tags on the ground. I'd be happy to pay $20 for a tag if it's one of our tags.
What Mexican states are they going to?
It's
Oaxaca?
Yeah. It's Michigan.
Michoacan?
Yeah. It's one of my bucket list things to do. I haven't been there. I have to go, obviously. Okay, so what's the point of this? So we've got a question mark here. So I told you that there's a group of people in Arizona, citizen scientists, that have been tagging monarchs for, I don't know, 5 years or so now. And they actually showed that, I mean, we didn't know where they'd go, but they go to Mexico too. So Western monarchs go to Mexico as well. And we presume that they come back, although we don't have tagging to prove that. But we do have tags, and just last week, 5 of them were found in Mexico that were tagged in Arizona last September. So there definitely is. And then if they go to Mexico, presumably they don't necessarily go back there. They probably could go back east. So, the point of this slide was that there used to be a divide here, or people thought there was a divide and the two populations didn't mix, but there likely is some interchange. And genetic work has shown that they're pretty much the same species, whether you get them in the east or the west. But up here, there's no lines up here, as you can see in Washington and Oregon. We don't know what happens up there. So that's the focus of this project. And so that's reiteration of what I just said. Flight of the Butterflies is the movie you should have seen. Hopefully you did see.
It's still playing.
Sorry?
It's still playing.
It's still playing. Okay, so it's come back again. So go and see it if you haven't seen it. And so much less is known about our population. Do they, the accepted theory is that they go to California. Do they all go to California, or do some go to Mexico? We say that because of Bob Pyle, and I hope you've read his book Chasing Monarchs, where he made observations on his trip from British Columbia down to Mexico, where he saw a lot of monarchs, they're vanishing bearings, they're disappearing into the distance. They were heading the wrong direction. They were heading southeast. They're in southern Washington. They were heading southeast instead of southwest. So, and he saw that in a number of places. So it set the idea up that maybe some do go to Mexico. And then, you know, with the Arizona work showing that if monarchs in Arizona do go to Mexico, and also actually some in Arizona also go to California. In fact, this year they tagged 2,000 and they had 6 turn up in California and 5 down in Mexico. So 50-50. Some years, apparently, they're all found in Mexico, none go in California. So maybe it's something to do with winds or whatever, but they have the opportunity or the potential to go in either direction. So what's to say that ours have the potential, from British Columbia, you know, to go that way or that way? And so that's the base, one of the major questions we want to answer in what we're doing with our monarch research. So there's very few records of wild tag monarchs migrating to California anyway. So the theory is based on like one individual. That came from Bob's work. Well, yeah, his work. It was a year after he did the book. He was doing some tagging down the gorge, and it turned up in Santa Cruz. I also made observations of them flying in different directions, and there were some releases by two school teachers in the 1970s and 80s, provided 20 recoveries in California. And that's been reported, but at least less reported is the fact that 2 of them were recovered in Utah, going in the other direction. So the Pacific Northwest is dominated by large farms, wild open spaces, and extensive forests, and relatively few people, as well as few monarchs. I mean, we see few monarchs. We have a very small human population compared to the east. So, you know, low populations of humans and monarchs in a big landscape means limited tagging. And even in the good years, it would have been limited. Now that we rarely see monarchs, it's almost impossible to have a good tagging program of wild monarchs. And it's ironic because milkweed is abundant in eastern Washington in some places, like this one here. But very few monarchs come up to use our milkweed. We have 2 species, and this one is actually found around Yakima and along. It's much rarer than the showy milkweed, but we do have it in some places, the gorge as well. So we have a lot of milkweed, but California, where the butterflies, if they mostly come from California, where they come from, their milkweed populations have declined dramatically during the past 3 decades through multiple reasons, drought perhaps particularly, but roadside strand. I mean, I've seen this myself. And the removal of milkweed from farmland. There was a lot of worry about it being toxic to livestock at one point, and so there's far fewer milkweed in California than there used to be. And populations of monarchs have declined, and this is a summary of overwintering colony counts in California from 1997 to 2013. So you can see there was a fall around the end of the century, and it hasn't really, you know, got any better. It got worse in 2007. Last year was slightly higher than the year before, but you know, it doesn't seem likely to spring up to there anytime soon. So you can see it's been quite a while now since we've had very large populations, when individual sites might have, you know, 100,000. This, you know, the total population was over 1 million in all the sites. So there's about 76 to 162 overwintering sites. So there's a lot of sites, and they're split between them all. And this shows the number of them all combined. So it's not a pretty picture, and the same applies to the eastern population too. We've been monitoring the news lately. You would have heard about the decline of the monarch and the fact that it's, you know, it's its lowest level ever. You know, they measure the population in size of hectares there, less than half a hectare, which is like 2 acres, or less than 1.5 acres or something. So it's tiny compared to what it was, you know, more than a decade ago, and it probably will rebound a bit because, you know, drought has had an impact too. But the main culprit is lack of milkweed again, destruction of milkweed, particularly in agricultural fields, by using genetically engineered soybean and other crops that allow herbicide to be used, and so every milkweed is eradicated. The crop doesn't get hurt by that. the herbicide. So there's far fewer milkweed out there than there used to be. So it's not a good story. So that's in eastern Washington, that's sort of typical habitat. That's Lower Crab Creek, which is the advantage. The Columbia River, which is just near the hills at the back there. And that's the sort of typical habitat you'll find them. And particularly this area, Lower Crab Creek, there's an abundance of milkweed. I've never seen so much milkweed in one location as this particular site. It really is literally acres of milkweed. If you go out there in early June, it's just fragrant. You know, the atmosphere, the air it's just full of milkweed. And this is actually a breeding population. It usually holds a breeding population. Again, it's mentioned in Bob Pyle's book. That's one of the reasons why I found the site. And I monitored the population there last year. So you can see, this is a zero line through there. As you see through June and July, there's very few, just one or two individuals. I went out there for a 3-hour search. And then the end of July, beginning of August, the population increased dramatically. Local breeding, this is the breeding population. So quite high during August, and then about the 27th of August, they all just disappeared. That's when they set off on their migration. So it is a breeding site, and we don't have many of them, I don't think. This one is pretty good. Horn Rapids near Benton City, usually there's usually a few breeding there, but not many. And most monarchs in eastern Washington are seen late in the summer because the populations are built up, like you saw in that graph, and they're moving south or southeast or southwest, whichever way they go. But so how the solution to the low natural populations to tag butterflies, like that population I was monitoring, I tagged, I think, 22 individuals. You know, it wasn't easy to catch monarch butterflies. So, you know, that sort of effort by one person sounds good, but there's not that many sites you could do that in. And so, even if we had an army of people, we wouldn't get more than 100 or 2 wild individuals tagged. So, mass rearing the butterfly seemed to be the solution to that, to low natural populations, to mass rear them and then to tag them and to release them. So we came up with the idea of doing this at Walla Walla Penitentiary. They were looking for a project to involve prisoners a couple of years ago that would help them take their mind off of being in prison, I suppose. They really just wake up in the morning and want something to do, want something to occupy their mind. And if it's something to do with the outdoors, that seemed to be better as well. So I had the idea of getting them involved in this, and it's been a successful program. Incarcerated Citizen Science, Understanding Migration of the Monarch Butterfly in the Pacific Northwest. It's received quite a lot of publicity, and it's had a lot of benefits to the prisoners involved. And we're making progress with research on the butterfly. And so we've done it for 2 years now, 2012 and 2013. And everything that they've done, they've done themselves. So it's very hard to take things into the prison. I mean, it was a while before they'd accept milkweed in the prison, because they were worried that then prisoners might smoke it or eat it or do something with it they shouldn't do. So they're very wary about what can come in. So they made their own cages. They made everything that they needed, and that was part of what they did, and part of their application, getting them to be interested in something. But last year they also grew milkweed in the prison, but they also had to collect some from outside, because if you've reared monarch caterpillars before, they eat a lot of milkweed. But the devotion they showed to caterpillar rearing was remarkable. Obviously, it's a cliche, but they had a lot of time on their hands, and so they spent much more time in sanitation than scientists do, or I do. You know, I try to rear monarchs, but there's a limit to how much time you can put into looking after the caterpillars. They spend day and night looking after them as they're allowed to. You know, and so because of that, disease was very low, and a high survival rate occurred. So they got more from, you know, more butterflies from the eggs that they were provided with. In fact, you know, these guys are all, they're not just in there for a couple of years. These are life prisoners. These are, well, I think the guy on the right may not be, but the other two were there for the rest of their lives. So these are long-term prisoners. And it was sort of weird being with them and seeing them worried to touch a butterfly wing in case they hurt it. So, you know, that was quite an eye-opener. And they really got involved in the metamorphosis, the change that they saw, and being quite mystical about it and comparing it to their lives and how they could change perhaps, or how they had changed, or whatever. But there's some deep thoughts going on that I was, you know, told about. And it, you know, just in 1 year or one season, just a few weeks, 6 weeks or whatever it was, it really affected some of these guys. And the guy on the right has actually moved to a lesser secure prison now, and he's taken a tertiary education course in natural science or something, based on, I think, you know, from the experience that he had here. So it can change people's lives, apparently. So even the tags were made in the penitentiary, which they're good big tags. They were probably too big, and they weren't as sticky as they should have been. But we still got results in the first year, as I'll show you in a minute. But we've moved now to Monarch watch tags, the tags they use in the East. But they made the tags themselves in the first year. So in 2012, our first year, they reared and we released, or they reared and released 2,581 monarchs, and we had 11 recovered, which is about 0.5%. And so if you look at the data from eastern US, the recovery rate is about 1% if you combine everything. So 0.5% we were down on that, but I was quite pleased with getting 11 back. And yes, right. So this is an overwintering colony in California, and you can see the circle there is around the tag butterfly. Unfortunately, it wasn't one of mine. It was one of the Arizona people's tags. But this was, you know, having the tag butterfly, having the clustered butterflies in California offers a good way of actually trying to retrieve your tags by taking pictures. In fact, the person that took this picture didn't know there was a tag butterfly until he got the picture home, photoshopped it, and found the tag. You know, and you can actually zoom in and read the tag. So, you know, so there's a lot of potential perhaps to get the tags back. But 4 of mine were recovered from distances greater than 50 miles from release point. So I had 11 recoveries, and only 4 of them were 50 miles or greater. And the longest distance was 600 miles from Yakima to Bolinas, which is just north of San Francisco. So that obviously went to California. But the other one was 450 miles, or the other notable one was 450 miles to Brigham City in Utah, which I'll show you a map. Totally different direction. And, you know, so for the first year, we got evidence supporting what Bob saw and Chasing Monarchs in his book, but, you know, some do apparently go that way, whereas some go that way. Just 2 data points doesn't tell the whole story. You know, it isn't definitive evidence, but it's certainly suggestive of something happening more than just them all going to California. So that's on the larger scale, and you can, so, you know, Arizona's down here, I guess, if my geography is right. So you can see it's on track to go down there if it continues going down that side of the Rockies. So we did the same thing again this year, last year, 2013, our new tags, which are very sticky. They'll last for 10 years probably, since the other ones, they're recovering in Mexico now, are that old, some of them. We released only 1,240. We had more problems with disease this year for some reason. I don't think it was the prisoner's fault. It wasn't larval disease, something to do with the egg. A lot of the eggs didn't hatch, so not quite sure what happened there. But the overall number was still large compared to the number we would be able to tag in the wild. But the surprising thing is we've had no recoveries to date, not a single one. Having the people in Mexico last week looking, had my fingers crossed, but nobody found a Washington Monarch. So to me, this is surprising. The first year we only had 0.5% compared to 1%, and in some parts of the eastern US it's far greater than 1% recovery rate. And this year, 0% to date. We're rapidly running out of time to get any recoveries. So I'm wondering if they really are mostly going to Mexico, and if they're taking that longer route there's obviously less chance for them to be recovered. You know, they're rarely caught in the autumn when they're moving. It's only when they're overwintering. So, you know, you wouldn't really expect them to be recovered too much on this route. They're going to be there if they get recovered. And, you know, the distance is so great, our numbers are relatively small, and so the odds of someone finding one there are probably much less. Whereas if they'd gone this way, we would have expected to see more, I would have thought. Particularly as we put the effort in, Tanya and the girls and I put the effort in to go down there last Thanksgiving and look for them. And this is Natural Bridges, and you can see all the people there. There's Annabella there. They're all looking up at Monarchs. Annabella's not, looking down. Give her time. So for 3 days we were down there. Santa Cruz, that's the place.
Santa Cruz.
Santa Cruz. Yes, thanks, Jess. So the population, there was about 3 or 4 sites we looked at there, and each had populations of 2,000 to 14,000 monarchs. So we estimated we looked at 20,000 monarchs over those days. We took hundreds of pictures and lots of video. Tanya had the video camera, you'll see a picture for her in a minute. Hundreds of images, video footage. This is Natural Bridges again. It's pretty spectacular. Well worth going to see. There's Tanya videotaping of the monarch on her back. But we didn't set that up, that just happened. And then, so we didn't see anything. I mean, I spent hours after we got back, days actually, looking through all the footage and didn't see anything in all the, except this picture. There's a tag in there, would you believe? It's up there, and this is a Pacific Grove. And normally Pacific Grove, if you've ever been there, you just wander down the path and the butterflies all hanging around you. But the day we went, they were in the neighbour's property, outside the fence. And so I had a zoom lens and zoom over, I don't know how many metres distance, to get this crummy shot. But this is the shot that's got a tag in. And so you can zoom in, that's the best I can get. It's definitely a tag. You know, there's a tag, you can even see words, but you can't read it, obviously. And is it one of ours? It looks like one of ours, but it could be one of the Arizona ones. Although for an Arizona one to go to Pacific Grove would be pretty unlikely. They've never gone that far before. And so when I saw this, I told them at Pacific Grove, please look and find, you know, there's a tag out there somewhere, but nobody saw it again. And so maybe it died or it flew off or whatever. But that's as close as we got to getting a tag this year. But you know, I can see it there now. It's definitely, there's an arrow in case you can't see it. And so this is where the Arizona group had, and I made this presentation before. I'd heard about their recoveries down there. So, you know, they went in 2 different directions, which is quite remarkable. That's much more divergent than our ones going in sort of a triangle shape. They're going in sort of 180 degrees different, almost. So most of theirs turned up at the colonies around Morro Bay, Goleta, San Luis Obispo, down there. They've never gone up further to Pacific Grove. But it is possible, I suppose, you know, just go a little bit further. We'll never know. So the future, we're going to continue this work. Again, this is work that I'm looking for funding for. Not easy to fund monarch butterfly research. There's very limited bodies that will fund such work. I'm looking at the National Science Foundation. I tried last year and almost got funded. They were very excited about the prisoner aspect of things. They thought, one reviewer thought it was the best proposal he'd ever seen in his life. Unfortunately, they had to send it to 2 other reviewers who didn't like it as much. So they got the balance. But I'm trying again. And if we do get money, then we'll be able to expand the research and have greater numbers. We need greater numbers than just 1,000 or 2. We could have other prisons involved, other penitentiaries. And we also want to look at some other questions like the induction of dormancy migration. It would surprise you to know that we still don't know for sure the exact cues, the environmental cues that cause initiation of reproductive dormancy and migration in monarch butterflies in North America. There's been one scientific paper done on the work which was inconclusive, which showed that photo period or day length had a role, but it didn't have a complete role. I did my PhD on the monarch in Australia, as you know, and part of my work was this, the induction of dormancy, and I've written papers on that and showed that the Australian population is induced by not by day lengths at all, but by temperature only, by low temperatures when they emerge. So I'm thinking it's a combination here of the temperature and the photo period of the day length. So if we get funding, I hope to be able to prove that as part of the project as well. So we're not going to give up on the wild tagging though. I mean, that population at Crab Creek is, with a group of people like you group of people, we could tag 100 in a day perhaps if we time it right. We tried to do it last year. We planned a bit late, and by the time our date came around, which is the end of August or early September, they'd all flown away. I wasn't anticipating them to fly away on the 27th. I mean, literally, they were there on the 27th and gone on the 30th. Between the 27th and 30th, they just en masse disappeared. But this year, we're going to plan for August the 23rd. So if any of you want to come along on August the 23rd, you'll be most welcome to help us tag monarchs at Lower Crab Creek, which is near Vantage. And as long as the butterflies are there, I'll let you know if they're there. If the population's as big as it was last year or bigger, then we would have a tagging day. And if we can tag 150, whatever, that'll be great. And so that's my presentation. And there's Annabella again, and this time she's looking down and she's looking for an egg or milkweed. So we'll get a trained, yeah. I thank Bob and my family and my technicians who unfortunately don't have a job anymore for the other work and all the viticulturists and prisoners. Thank you for listening.
I mentioned there's evidently some interest in doing outside things in penitentiaries. I was asked to do a butterfly program for the Purdy Women's Penitentiary, and they immediately after the program asked if they could plant a butterfly garden on the prison grounds, and I think they're doing it this year.
Right, that's good. Yeah. Any questions on the Monarch?
The eggs for the Monarch, where do you get the eggs? Are they all Northwest Monarch eggs, or how do you do? That was a
You
lot.
mean to supply the eggs for the prisoners? Yeah, I missed that bit, didn't I?
2,000 eggs.
Yeah,
That's-
what I did, I've got a permit from the Department of Fish and Wildlife to actually catch monarchs and to get them to lay eggs and to rear them. I mean, apparently shouldn't be telling WBA members you're not actually allowed to catch butterflies in Washington State. The Fish and Wildlife Department knew about it. Well, obviously they do know about it, but it's a law that you're not allowed to catch butterflies. And so with this high-profile project, publicity and everything, the first year I didn't have a permit, so I had to get a permit. And so when I got a permit, I'm allowed to collect female monarchs and get them to lay eggs, and then I gave the eggs to the penitentiary. But each female will lay, anything up to 800 eggs or more. So a single female, young gravid female, will lay a lot of eggs. So you don't need many females to get the numbers that we produce.
And you start with wild eggs every year, right?
Yes. Well, that's we don't want to have eggs from elsewhere. Bob Pyle is very, very concerned about this, as you know, about transporting monarchs across state lines and using monarchs that aren't here in the first place. So it's very important that we only use monarchs that are here, the progeny of monarchs that are already here, not monarchs that come from elsewhere.
You go out and catch new butterflies in the wild for eggs each year?
Yes.
Okay.
Yes. Yeah. So there's no continual breeding.
Okay.
It starts new again every year. And so it's dependent on whether we see any monarchs in. So yeah.
So I was in California a couple of years ago at a vineyard and noticed that they were allowing wild mustard to grow between the rows and quite expensive.
Yeah.
Is there any, I don't know, that's probably not native to Washington, but Does that have any beneficial aspects to any insects or?
It probably does, but as you say, it's not native. It's not even native to California, I don't think. I think it's an exotic mustard. There's a whole range of so-called ground cover or companion plants that California viticulturists use that have sometimes have an insect attractive purpose, but they have to be irrigated, or they only last for short, like the mustard, for a short period of time. And so that's where the work has been centred in the past, on plants that are not native and not going to survive the long term and have to be resown each year. And growers don't want to do that. It's extra work.